So let me pull that up. If you guys want to give a guess about our question for our warm-up. So what kind of algae bloom produces bright blue lights in the water? So if you have a guess, you can go ahead and throw it in the chat for us, and we'll get started right at 6 as more people start trickling in. I like that guess, phosphorescence, good. So I noticed we had a few more people come in. We are going to get started right at 6 o'clock, but I did throw a question in the chat if you guys want to kind of get your brain in the HABS spirit today. So what kind of algae bloom produces bright blue lights in the water? And you can take a guess if you'd like in the chat. Or if you've even seen one, that'd be kind of cool. Seen them on the York River, awesome. I heard there was some pretty good ones on the York River this past summer for about a couple weeks, some really bright ones. Okay, guys, so we're going to probably have a couple more people trickle in, but it is 6 o'clock, so I do want to go ahead and get started. So welcome to our April Discovery Lab, Harmful Algae Blooms, or HABs for short. And the answer to our question this evening, what kind of algae bloom produces bright blue lights in the water? We know those as those bioluminescent blooms caused by all those dinoflagellates. So you see those a lot during the more warmer times of the year. If you've never gotten the chance to see one, I really hope you do, because they are pretty remarkable when you get the opportunity to see those. But we're going to go ahead and get started this evening. So we do have a little update for you guys. So this is our last Discovery Lab for the beginning of this year. We have been discussing possibly doing some fall Discovery Labs, whether they're virtual or in person. That is still up for debate at the moment. We kind of have to wait for VIMS to kind of make that call about campus reopening, but we would really like to do some fall opportunities for Discovery Labs this year. And we've come up with a list of a few possible topics, but we do also want to get your input on whether you like one of these possible topics, or if there's a topic maybe that we haven't thought of that you'd be interested in hearing about. So I'm actually going to launch a poll, and it should pop up on your screen. And so you can go ahead and answer, it's anonymous, about maybe one of these that you're really interested in, or that other option. If there isn't one listed here, you can throw in the chat or the Q&A maybe an idea that you think would be a really cool Discovery Lab, because we love getting people's inputs, because sometimes you guys come up with some really good ideas that we haven't even thought of before. So we would love to definitely get your feedback on these possible topics, or any ideas that you guys have as well. Awesome, we have a lot coming in. Everyone's really close, pretty even split. I'll give you another couple seconds. Okay. So I'm going to go ahead and end that poll. So you might be able to, I can share the results for you guys, but these are some of the top options. It's pretty split all the way. So we'll definitely take note of these ideas that you guys, or if you throw those ideas in the chat, we definitely want to record those. And we'll keep you guys posted on what's going to be happening later on this year with our Discovery Labs. And so to get those updates, you guys can definitely follow us on our social media platforms. So our Facebook, many of you guys might know that's where we post a lot of the updates for our educational events. Also our Instagram and Twitter will be posting there. So we'll definitely make an announcement about whether those Discovery Labs are going to be live or in person, and we'll give you those dates as well. But jumping into today's Discovery Lab, we have Sophia and Maggie with us. They've been with us for each Discovery Lab, if you guys have been with us as well. So they are eco ambassadors. So they are both students over at William and Mary, and they have been assisting us in creating a bunch of resources for all the topics this past year. And this is kind of their final run through. So they're going to be doing the majority of this lab this evening, doing the demonstration. We're going to do our Kahoot game as well, but we're excited to have them kind of lead the pack towards the end of our Discovery Lab. But again, they're both juniors over at William and Mary, majoring in biology, environmental science, and anthropology and history. And they've been doing a great job in creating some really wonderful resources for us. So you'll meet them a little later. And the outline for today's Discovery Lab, we have Marta Sanderson with us. So she is our guest speaker this evening. She'll give her presentation, and you guys can ask questions in the chat, or we would actually prefer them in the Q&A section. So in your little toolbar on Zoom, you should see the Q&A. So if you have some questions, you can definitely pop them in there, and Marta will have some time at the end of her talk to answer a few of those questions that you guys ask. And then we'll have Maggie and Sophia taking over. They'll be demonstrating the Enviroscape, and they'll be doing that. They filmed a video to show you guys and talk a little bit about how that relates to HABs. And of course, we're wrapping up this evening with our trivia, HABs trivia through Kahoot. So if you've been with us before and you're familiar with that, we got another one coming. And then you'll get an email too, either later tonight or tomorrow, about some additional resources that they created that you guys have the opportunity to explore after the Discovery Lab. And on that note, I do want to introduce our lovely guest speaker, Marta Sanderson. So she is a faculty member over at VIMS. She's a laboratory manager in the HABs and Toxins Lab. And so she's going to be taking over and speaking to you a little bit about HABs in general and what they are, and then some of the research that she does over in her lab at VIMS. And so with that, I'm going to go ahead and stop sharing my screen, and I'll turn it over to Marta.
Let's see if I can get this to start.
It's always the tricky part.
Okay, it doesn't seem to be showing up. Can you guys see my screen?
Yep, we can see it.
Okay. All right. Well, thank you, Tara, for that introduction, and thanks to everyone who's joined us this evening for a virtual Discovery Lab to talk about harmful algal blooms and toxins. So let's jump right in. What are harmful algal blooms, or HABs, as I'll be referring to them throughout this talk? The first thing that might pop into your mind may be a red tide, and that would be correct. That is a common term that's used to describe these, let me get my laser pointer. It's a common term that's used to describe some of the better known HABs, and the name comes from the red color that the water turns when a specific species of algae blooms in that water. However, HABs are not always red, and they're not associated with tides. They can be brown or green, as shown in these pictures, And the color just depends on the species of phytoplankton or algae that's actually blooming. They can occur in all types of water, from freshwater to seawater, and they're found all over the world, mostly along the coastlines and in inland bodies of water. And they're becoming more widespread and more frequent due to climate change. But before I go any further talking about HABs, first let's take a step back and talk a little bit about algae. So algae, or phytoplankton, are the tiny microscopic plants that are found in all bodies of water. They are unicellular or single-celled, and they come in all different shapes and sizes, as you can see in these images that I have up here. They can be quite beautiful and intricate, and some of them like to form chains like the ones shown here. Like this one is a chain of 9 individuals. This one is a colony. The individual cells stick to a mucous membrane and form a colony. So they come in different shapes and sizes, and I think they're really cool to look at. But they're very important. Phytoplankton are very important because they are primary producers. In other words, they can make their own food through a process known as photosynthesis, just like land plants do. In the case of phytoplankton, they use sunlight and convert carbon dioxide that's dissolved in the water to simple sugars, which is their source of food, and they give off oxygen as a byproduct. An interesting aside would be that roughly 70% of the world's oxygen is produced by phytoplankton. That's a lot of oxygen coming from these tiny microscopic plants. So because they don't rely on any other organism as a food source, they are considered the base of the aquatic food chain. Simply put, the phytoplankton here, they grow and they produce their own, they make their own food. They are then consumed by zooplankton and small fish, which are then later consumed by larger fish and so on up through the food chain. Also shown in this cartoon are some shellfish here, and they are filter feeders. They pull the water through their mantle and extract or pull out the phytoplankton and use that as a food source. So hopefully I've shown you briefly that not only are phytoplankton beautiful, but they're also very important marine organisms. So phytoplankton are a good thing, right? Yeah, they are. However, sometimes too much of a good thing can be bad, and that's where HABs can come into play. So exactly what is a HAB, and why should we care about them? Well, first let's start with an algal bloom. Blooms like the one shown in this picture are a result of a rapid increase in the number of phytoplankton cells of the same species in the water. We usually consider it a bloom if there are more than 1,000 cells per mil of water. That's 1,000 of those individual cells in about a quarter teaspoon of water. Now, all algal blooms are not harmful. In order to be considered a harmful algal bloom, they have to have a negative impact on either the environment, the economy, marine life, and/or human health. So some of the causes or environmental drivers of HABs are listed here. One of the main factors for bloom formation is an increase in the available amount of nutrients. So phytoplankton need nutrients like nitrate and phosphate to grow, just like a land plant does. And they can get these extra nutrients from agricultural and residential runoff. For example, fertilizers, which are very rich in nutrients, are put on crops to enhance growth, or we can put them on our yards to make our grass grow nice and green. Well, these fertilizers can run off of the land into the watershed during a rain or wind event and eventually end up in our waters, providing that extra source of nutrients that are needed for the rapid algae growth. Excess nutrients can also be introduced through sewage discharge or urban and industrial runoff. Other factors that may cause blooms to form are related to climate change, such as warmer water temperatures or changes in the pH or salinity of the water. And as I mentioned earlier, as we experience climate change, we are seeing more extensive, longer-lived, and more frequent harmful algal blooms. So some of the negative effects associated with these blooms, as you saw in some of the earlier pictures, the water can be discolored or have a scum on the surface of the water. So imagine you're going to go to your favorite beach for a swim and you get there and the water's kind of a brownish red color, or maybe you are going to go fishing to your favorite lake. And when you show up, there's a bright green layer of scum on the water. You might decide that you don't want to swim or fish in those waters. So these negative impacts don't just ruin your day, they can also affect tourism and the economies of an area. In addition, you can imagine that very dense blooms can block sunlight from penetrating down through the water to the areas below the bloom, and therefore causing light limitation to some of the animals or plants that are underneath the bloom. Along those same lines, as a bloom dies off, it starts to decay, and that uses up oxygen, and areas of either low or no oxygen can form. And this is also detrimental to many forms of marine life. So, and the last thing I have listed here is toxin production. Certain HAB species can produce toxins. Not all of them do. And then those that do don't always produce toxins either. And I'll talk a little more about toxins because that's part of what we study in our lab. So toxins can have complex structures like those shown here, and they can be released into the environment, which we refer to as extracellular toxins, or toxins that are found outside of the phytoplankton cell. Or they can remain inside the cell, and we refer to those as intracellular. Now, I changed the color of my phytoplankton from green to red to indicate that they now contain toxin. And next, I'm going to show you how toxin can be transferred up the food chain. So during blooms, these toxins can move up the food chain by zooplankton or small fish and filter feeders eating the toxic phytoplankton and accumulating these toxins in their flesh, and then another organism coming up and eating them, and the toxins are now accumulating in that animal's tissue. This is a form of trophic transfer for toxins. As you can imagine, aquatic animals can experience various health issues from exposures to HABs. These include things like physical damage. When a fish swims through a very thick, dense bloom, it could get its gills clogged, or if the gills or skin can be eroded swimming through these blooms. You can also see things like muscular damage, which will cause problems with mobility, or neurological damage. Often if you see or hear about a mammal stranding or something like that, it can often be traced back to a harmful algal bloom that the toxins have eventually made its way to that mammal, causing some disorientation and resulting in that stranding. We can also see slow or halted growth, and in some cases, mortality or death. So those are some of the things that marine life may experience due to HABs. Here are some of the ways that humans may be affected. One of the ways is by accidental consumption of seafood that's contaminated with HAB toxins. This table lists some of the main syndromes that are associated with eating such contaminated seafood products. I'm not going to go into a lot of detail on the different syndromes and what they cause, but they do exist. And some of the other health issues that are associated with HABs include exposure to cyanotoxins through drinking water, or dermal exposures and respiratory issues from either recreational water use or working on the water. But don't worry, our local, state, and federal governments, the Departments of Health, Environmental Quality, EPA, FDA, things like that, as well as HAB researchers, work together to monitor for HABs and to implement regulations and guidelines on drinking water, shellfish harvesting, aquaculture production, as well as inspecting seafood before it goes to market. If an area is deemed not safe, signage is posted to alert the public, as shown in some of these examples here. Okay, so in Chesapeake Bay, we have over 37 species of harmful algae. Fortunately, we have not had any reports of any of those major human health syndromes from anyone eating shellfish in our area. However, there have been some reports of commercial fishermen who are working either in or near very dense blooms have reported dermal issues and respiratory issues like irritated, itchy skin, or maybe a cough or a sore throat type of thing. Now, if you ever want to check out and see if there is a bloom occurring in our state, the Virginia Department of Health has a HAB surveillance map, and they update this map from May through October. It's an interactive map, and you can see where any bloom is occurring within the state of Virginia. So I have taken a screenshot here of September of last year, and I focused on an area right off of VIMS, and you can see all these little brown dots are areas of a bloom. And if you follow down here and look, the brown means that this is an ongoing bloom and that there is no HAB advisory issued. If you were to click on a specific bloom, this dialog box would pop up and it will tell you where the location is, the date, the species that's blooming, as well as the concentration of that species. So if they have that data, this one was over 300,000 cells per mil. It was a pretty dense bloom. And it also tells you important information as to whether or not this bloom is associated with any health issues to either human or marine life. So now I want to focus on some of the main HAB species that may be present or that are present in Virginia waters and that may show up on that interactive map if you so choose to go and check out any of the blooms. So in early September, we might see Dinophysis species. Let me start this video. Dinophysis are dinoflagellates, and they have this whip-like structure called a flagellum, which allows them to spin and move around in the water, as you see in this video. This group produces ocadaic acid, dinophysis toxin, and pectinotoxins. And these toxins are associated with diuretic shellfish poisoning, which is one of those human health syndromes. Next, we have two more dinoflagellates, Carlidinium vinificum, which produce carlotoxins, and Prorocentrum minimum. And these two often occur at the same time, and they are both considered harmful algal species because they have detrimental effects on some marine organisms. Then we also have a diatom, which is Pseudonychia species. And they produce a toxin called domoic acid, and it is associated with another one of the human health syndromes called amnesic shellfish poisoning. These four groups usually show up in the early spring and into summer, and some of them may form some small short-lived blooms. It's not until we move into midsummer that we tend to see these much larger, more visible blooms of a dinoflagellate. I'll start that video. So and this particular species is called Margalephidinium polycocoides. It usually starts to bloom in early July to mid-July. And prolonged exposure to this HAB can be harmful to larval shellfish and finfish. And it's one that forms chains. As you can see, there are a couple of individual cells attached here to each other. So it is a chain forming dinoflagellate. It is typically followed by another dinoflagellate called Alexandrium monolatum. The toxin associated with Alexandrium monolatum is Goniodomin, or Goniodomin A, or goniodomins, and there has been mortality of oysters and oyster larvae associated with these blooms. It's also a chain former, as you can see in the video of them moving around in the water, and it's one of the ones that produce the spectacular shows of bioluminescence at night. So if you haven't seen one, I highly recommend, if you hear that we're having a bloom of Alexandrium monolatum, typically I would say late August, early September, You should try and get out to the York River area and see if you can see that, because it's spectacular. Okay, so now that you know about some of the HABs and the toxins that we find in our air, let's talk about the research that my lab conducts to better understand HAB ecology and toxin production. So we do this by conducting experiments in the lab. We can design culture experiments to investigate how different environmental factors play a role in bloom formation and toxin production. So for example, we can take a culture of a specific harmful algal species, grow it up in the lab, and then manipulate how much nitrogen or phosphorus, what forms they get. We can change the light, the temperature, the pH, any different variable, and then determine how well they continue to grow or whether or not they are producing toxins. We can research each one of these drivers on their own, or we can research multiple of them at the same time, which is what you would more likely see in nature. In addition, we also conduct bioassays, and that allows us to look at how HAB species and toxins may affect aquatic organisms. One example is where we would combine oyster larvae, shown here, with either a single HAB species or multiple HAB species that occur at the same time, and then observe the effect on the growth or survivability of those baby oysters. These experiments can be carried out in small scale, like in a 1 ml well plate shown here, or in larger bottles or jars shown in this picture. These types of bioassays can also be done with different shellfish and also with baby finfish. Now, we do a lot of our research in the lab because we can carefully control each one of those factors or drivers, but we also do research out in the field. And I want to highlight a couple of our research projects that have field components. Okay. First, we have CB-Tox, which was designed to look at HAB and toxin formation in the lower Chesapeake Bay over space and time. So we chose 12 different sites in the Virginia portion of the Chesapeake Bay, and we sampled at those sites roughly every 2 weeks for over a year. And we did this so that we could get a baseline understanding of what HABs are present and what toxins are being produced throughout the year. And we did find that some toxins can be present at low concentrations year-round, even without a bloom happening. At 4 of those sites, we also deployed oysters, and here's 2 students deploying an oyster cage, to look at toxin accumulation and detoxification in oyster tissue. And we work with shellfish hatcheries and aquaculture facilities to investigate ways to minimize the effects of HABs and toxins in their facilities and on aquaculture production. Whether we're working in the lab or the field, we process and analyze a lot of water. We collect whole water or grab samples, which give us a snapshot of what's present in the water at a very specific time. Some of the types of samples are shown here. With the cell ID, we can use a microscope to help us identify the HAB species in the water and then count how many number of those cells are there. The remaining samples we use to measure how much toxin is in either the water or the cells themselves. And we also can do this for the edible portion of animal tissue. For the toxin samples, we use a combination of both chemical and physical methods to extract those toxins from the seawater or the phytoplankton cells in order to get at the amount of toxin that's present. So here are some of the tools that we use to analyze those samples. As I said, we use microscopes to help us identify and count the cells. Then we can use various instruments and methods to extract the toxins out of the water or the animal tissue. And I'm not going to go into too much detail on that, but we do this so that we can eventually quantify or determine the concentration of those toxins using our ultra performance liquid chromatography tandem mass spectrometer shown in this picture. Then we also collect another type of sample, which is different than our whole water samples. Our whole water give us that amount of toxin or species that's present at a very specific time. These samples that we call SPATs, it stands for solid phase absorption toxin tracking, or SPAT. They give us a picture of the dissolved or extracellular toxins that are in the water cumulative over a time period. So we often deploy these for 2 weeks at a time, and we're able to tell what toxins were present in the water during that time period, as opposed to what happened to be there the day we sampled. And then I want to highlight another tool that we use in our HAB research, and this is the Imaging Flow Cytobot, or IFCB. This instrument is basically an underwater microscope, and it takes high quality images of phytoplankton that you can see here that are in the water. We have ours deployed off of the VIMS campus, and it transmits its data back to our lab on an hourly basis. That data is then run through classifiers that we have built using computer algorithms that separate all these images into different phytoplankton groups or zooplankton groups. So we have roughly 40 phytoplankton groups and 15 zooplankton groups that we currently use in our classification systems. And from that data, we can generate things like these weekly reports that focus on either one species, or in this case, we were looking at 3 different species, and have those reports generated and sent back to us. And this allows us to use the IFCB as an early warning system for a bloom, because we can start seeing the numbers of a particular species start tick up as the bloom starts forming. And then we can use the data for other purposes as well, but I think I will stop with that. And I hope that I've shown you some of the ways that our lab is working towards understanding HAB ecology and toxin production. So thank you very much. And I guess if there are questions, we can take some questions.
Awesome. Thank you so much for speaking with us and giving everyone a wealth of knowledge about these events and how they form and how you guys are working with them. But we do have a couple questions that came through for you to answer if you have some time. So someone's asking about, are there any kind of methods that exist to eliminate these blooms once they've started or they've occurred, or do you just kind of let them do their thing?
Well, there are for certain species, but not for all. And currently in our area, we do not have any methods that we use to sort of stop them once they start. But I do know it is an active area of research, and there are people who do study using different techniques to mitigate those blooms.
And then someone earlier was talking about they've seen the signage that's been placed at, rivers where it's saying it's not safe to swim, things of that nature. And they're wondering if that has any kind of, would it have any negative effects on, say, like their dog, if they're walking their dog along that area, would it cause any harm to their animals?
So yes, it can. Just like if a human is exposed to some of these toxins and we have symptoms from that exposure, your animal can also have similar symptoms. And there have been cases reported, horrible cases, where animals have actually, like pets, have actually died from swimming in the ones that come to mind are microcystis blooms. They bloom in fresh water, and some of these happened just last year in Florida.
And then someone's wanted to get a little more elaboration on exactly how small these plankton are, the cells that you work with.
Okay, so they are microscopic. They are tiny. You can't see them without using a microscope. And typically they can range from like 20 to 40 microns. Some can get bigger if they form chains. So when they form a chain, you can see the chains sometimes If there are enough individuals attached to each other, you can see the chain without using a microscope. But typically you need a microscope in order to see them. And we're talking like typically 20 to 40 microns. They can be bigger as well, but that's smaller than like the diameter of your hair.
Yeah. So the best way to see those are definitely those, I love those videos that you showed. Those are always great to really see them under the microscope for sure.
Yeah, and they are available on the VIMS website. You can go to HAB videos and there's a couple of different ones you can see.
Great. And someone's also asking about citizen science projects. Are there any kind of citizen science projects going on with HABs? Is there a way for the public to kind of get involved with this?
So yeah, there's always ways to kind of get yourself involved. Obviously, if you see an area that looks discolored and you think it might be a harmful algal bloom, there are like the HAB hotline that can, I don't know the phone number off the top of my head, but there is a HAB hotline that you can call and report that you think there may be a bloom happening and give the location and all of that, and someone will go check that out. Or if you see things like fish kills or animal strandings or things like that, obviously you can help point those out to the Virginia Department of Health, and VIMS also works heavily with this HAB hotline.
Yeah. And that kind of transitions really well into this next question about, we talked about the signage that can be found, but say there's a place where there's not signage out and people are unsure if they should swim in the water. You know, what are some of the signs out there that maybe that's not safe to go in, even if there's not signage? And I know you talked a little bit about, well, if you see fish floating on the surface, probably not a good time to go swimming.
Yeah, so that's a much more difficult question to answer, because sometimes you may not be able to tell if the water is actually colored because of a phytoplankton bloom, or if it's just we've had a lot of wind and the bottom's been kicked up, and so there's a lot of debris in the water or turbidity, and that may make you think it's a bloom when it's not. So I would suggest one way would be to check out the HAB surveillance map before you go somewhere. But yeah, unless you really can tell that it's not due to dirt or silt being kicked up in the water, there's no way for certain to say, yeah, I can't swim here. Now, in our area, like I said, we've had very few reports of issues for human health from any of these HABs. But, you know, at the same time, if you're unsure, maybe it's better to stay out.
Yeah. And then I think we have time for a couple more. Someone was asking about, why are only some blooms considered toxic?
Okay, so if you have a bloom that occurs, but then there's no actual negative impact to either the economy, the environment, or any marine life, or humans, of course, then it's not considered a harmful algal bloom. So you can have blooms that occur like in the open ocean, we have these blooms that occur every year. It's like a succession of phytoplankton from one species to the next, but it's not causing any sort of negative effects. And those blooms are not considered harmful. The phytoplankton that are blooming are not producing any toxin either. And as I said before, I hope I mentioned that all harmful algal species do not produce toxins. But obviously, if they did, they would be in that category as harmful. So,
do you see, tend to be less blooms that are less harmful, or sorry, do you see more harmful blooms or less?
Well, I see more harmful because that's what I'm looking for. But I'm sure there are quite a few other blooms that are not considered harmful. And they don't necessarily change the water color either. You can still have a lot of biomass without that color change.
So we have time for one more quick question. And they're asking, in the tidewater area, are we only seeing blooms in saltwater environments or only freshwater, or both?
So you actually can get them in both. And I focused mostly, well, all when I showed you the HAB species in Virginia was all marine species. But we do get blooms in lakes and ponds, and it's typically microcystis that's blooming, and you can see a green color usually in the water with those. So we do have the freshwater blooms as well. And some of those actually, kind of cross over into the marine realm and are in the brackish waters. And microcystis is one of those that does that. We've seen both marine and fresh blooms.
So we are going to continue on. Those are great questions. And again, we want to thank you for joining us this evening, being our guest speaker. If you guys have any further questions, feel free to ask those. Marta, if you're comfortable with sharing your email, if people have further questions about some of the projects you do or just HABs in general, if you don't mind putting that in the chat, but
yeah,
totally
I can do that.
But I know we can't all see each other, but we'll give you a round of applause. And thank you again so much. And we do want to go ahead and turn it over to our eco ambassadors. So Sophia and Maggie are going to share a couple of the projects that they have set up for us this evening, along with our Kahoot game as well. And I noticed someone had their hand raised, so if you want to throw us a private message, if you have a question, you can do that in the chat for us. But I'll go ahead and turn it over to Sophia and Maggie.
Yeah, hi. Thank you so much to Dr. Sanderson for her wonderful presentation. We're going to follow up with kind of an exploration of, we learned about how one of the major causes of harmful algal blooms is nutrient pollution, that having too many nutrients in the water can cause these blooms. So we're going to do a little demo. We videoed a demo that will kind of explain how a lot of those pollutants get into the water. I'm going to share my screen. Here we go. There we go. So the one thing that I want to clarify before I start the video is that we're going to be talking about the Enviroscape as a watershed area. And a watershed is an area of land and all of the rain that falls on that area and all of the streams in that area all drain to a common outlet, like the outflow of a reservoir, the mouth of a bay, or any other kind of opening to the ocean. So the Chesapeake Bay area is a really large watershed. It covers about 6 states and drains millions of gallons of water every year. It's the second largest in the United States, second only to the Mississippi, into the Gulf of Mexico, which drains almost the entire Midwest. It's huge. So I will get the video started for you all. So this is our Enviroscape that represents our watershed area, and it's got a bunch of different zones on it that serve different purposes. So we're going to be putting different materials on it to represent different pollutants, and then simulating a rainstorm and seeing what happens. So our first zone is the agricultural zone. Farming.
Maggie, are you able to rewind a little bit? I know the sound went out a little and it's a little low right now.
On there absolutely, just a moment. And then these sprinkles. And I can see if I can change the volume as well. And seeing is that any better? So we've got ground oats to represent fertilizer.
Yeah, that should work.
We've got this green Kool-Aid to represent pesticides, these sprinkles to represent trash, and these coffee grounds to represent sediment. So our first zone is the agricultural zone. Farming tills the soil and makes it loose, so you get a lot of excess sediment. So we're going to be putting a lot of coffee grounds on there to represent that. And then another important part of farming is fertilizer and pesticides to keep the crops healthy. So we're going to be putting some of those on as well. Our next zone is the construction zone. Construction digs up the soil and makes it really loose, so you also get a lot of loose sediment there. So we're going to put some down there. Next is the neighborhood. People use a lot of pesticides and fertilizers to keep their lawns nice and green, so we'll be putting some of that. As well as trash that comes out of everyone's houses that eventually gets washed into storm drains and such. Next, we'll be looking at the resort. So what you get really commonly is trash on beaches that are popular. So we're going to be putting a bunch of trash on this beach because people will leave their trash. As well as on the golf course, again, people use a bunch of fertilizer and pesticides to keep it nice and green and good for playing golf. So we're going to put a bunch on there. So now we've got our rainstorm, and as we all know, the Chesapeake Bay Area is very partial to heavy rain. So this is primarily how things that we think of as land pollutants end up in the water. So let's start with our agricultural area. Here we go. Starts raining. You can see the Kool-Aid from our pesticides is running down through our river. Some of the sediment is also washing down. Our oats seem to be a little heavy, but they're also making their way down into the waterway. If we go over to our construction site, the sediment also starts moving down. Some of it gets wet and gets stuck to the bottom, but a lot of it is moving all the way down into our ocean. As we move to our neighborhood, the same thing happens. You can see it start to collect in low areas, and then once it fills up, it starts to drain down into the ocean again. On our beach, our trash is a little heavier, so it doesn't quite get moved, but it's a little colorful. But that primarily stays where it is. And then back to our golf course with all of our pesticides and our fertilizers, you can see that's also washing down into the bay. So you can see stuff that starts up high, drains down, lower into the watershed, and that all of these smaller rivers and estuaries eventually drain into the bay. After our milder first storm, a second stronger storm comes and hits our watershed. You can see all those heavy materials like sediment and pesticides and fertilizers and trash is now far more easily washed into the watershed. You can see the transference from those smaller rivers and streams into the marina and then out into the ocean. So as you can see, as our second part of the storm is coming through, the little more violent part, even more of our heavier materials like our oats and our sediments and our trash has been washed all the way down into the ocean. Stuff from the construction site has run down these nice paved streets down into the marina, into the estuary, and then into the ocean. Pavement can really help water and therefore things carried in stormwater drain down into waterways. So when you see in the ocean, you can see all of our sediment, our trash starting to dissolve. Our water is nice and blue from all of the pesticides. And you can see little bits of oat in there as well. You can see that even though you might think of many of these things as pollutants that go on land, that you haven't put directly into the water, through rain and other sort of processes, they all end up in the bay anyway. So many of the things that we do in our everyday lives that we might not think about affecting the ocean can affect the ocean pretty seriously. You can see our water is looking pretty polluted down there. And this can cause a lot of problems with fish, but in our case, all of these pollutants contribute to causing harmful algal blooms.
Great. So thank you for watching that little video. Basically, the most common one of the pollutants we mentioned that contributes to nutrient pollution would be fertilizers, because all those nutrients that are great for making your lawn grow also help HABs grow. We see that the increase in both kind of climate change that Dr. Sanderson was talking about, and then also nutrient pollution cause way more HABs that affect our everyday lives. And so now I will turn over to Sophia, who has a Kahoot for us all to do.
Yeah, and thank you guys. That was a great demonstration about how even though you might live, very far away from the actual coastline, our impacts can definitely be seen in those local waterways. So if you guys have questions about that demonstration, the Enviroscape in general, you can feel free to ask. But on that note, we will turn it over to Sophia, and she'll be leading our Kahoot trivia game to wrap up the evening.
Thank you, Maggie, and thank you, Tara. I'm going to share my screen so you guys can see the pin. So if you guys go to www.kahoot.it right here, you can log in, and then you'll type in the game pin number, and we can get started.
Yep, and you guys can use a mobile device, iPad, another screen to enter your name with the Kahoot game. And also, Sophia, while people are entering the names, do you mind, in case there are people who haven't played Kahoot before, if you can explain how it works for them?
Yeah, sure. So what's going to happen is on the screen where you're watching the Discovery Lab, if it's a computer, if you have it plugged up to your TV, that's where the questions are going to appear. So you'll see the question, and then you'll see the answer choices with little boxes, and they're probably going to be different colors and shapes. And those are the same answer choices that you're going to see on your device where you're logged into the Kahoot. And so while the questions appear on the screen, you're actually going to answer the questions on your individual device. And that's how you participate in the game. It becomes a little clearer once we do the first question. Okay, so we'll give everyone a little more time to log in. And we're also going to do a prize pack for the winners. So I think we went over a lot of the material during the Q&A portion, and also during the demo and Dr. Sanderson's presentation. So hopefully we got a lot of these right, and you guys are really pumped about it. All right, we can go ahead and get started. Oh, okay. All right, here we go. Okay, so the first question is going to be, where do HABs occur? So just a reminder, you can see the answer choices on your big screen, but on your separate device, that's where you're going to answer the questions. Wow, good job, guys. We got them all right. HABs do occur in all types of water. All right, Rory's at the top of the board. All right, so the second question is, what do HABs stand for? Harmful algal blooms. All right, people moving up. Just as a reminder, if you answer it correctly, you get points, and also if you answer quickly, you get more points. So what % of states report HABs annually? All right. So you wouldn't think that this was a problem across the country, but actually a lot of states do report HABs every year. All right, we have a true false question, or just a which of the following is false? All right, good job, guys. So we know that HABs do have a negative impact on humans as well as other organisms that live in the water. All right, Caitlin has a streak with 4 correct answers in a row. That's awesome. All right, so we know HABs are caused by both plankton and nutrients as well as nitrogen and phosphorus that are in the water. Now we have a true false question. Is it bad to have algae in the Chesapeake Bay? So as you guys know, there is good algae, not all algae is bad, but when you have too much, then you can have harmful algal blooms, and those can be really impactful. Okay, good job, guys. We got this one all right. Global warming does increase the intensity and the likelihood of harmful algal blooms in the water. Rory still in number 1. And we have our last question. Excess nutrients mainly enter the bay in what form? All right, good job, guys. So agriculture is the main cause of excess nutrients in the bay. These are our final standings. In 3rd place, we have Gabby. Good job. 2nd place is Dallas. And in 1st place, is it going to be? We have Rory. Good job, guys.
So if Rory, you want to put your, you can private message either Sarah and I your email address, and we can contact you about that prize pack. But you guys did really well for that Kahoot game. So kudos to you guys. You were paying attention, or you are just experts in HABs. But I do want to thank you guys so much for attending this evening. If you guys have any other questions, feel free to ask those. We'll stay on for a couple more minutes. And again, this is our last Discovery Lab until like the summer, because we're going to be getting into some of our online summer programming. But again, we might be doing some fall discovery lab. So make sure to follow us on our social media platforms for those updates. We will definitely post there. And again, we want to give a shout out to Marta for being our guest speaker, and for Maggie and Sophia for doing some great work tonight, and for all of our other discovery labs. But I hope you guys have a great rest of your week and evening, and hopefully we'll see you in the fall. So thank you guys.